Hello everyone, welcome to the online lecture for Chapter 11. In the previous lectures that we've been dealing with, we've been working on understanding neural function. That is to say, input, decision, action potential, neurotransmitter secretion, and so forth. Well, we're going to apply that in the efferent division, if you will, specifically in the autonomic neurons. I will use somatic motor neurons as an example, but primarily what we're going to do is focus on the autonomic neurons, and it's sort of summarized down here in this picture. We're going to be using a picture like this quite often in this lecture. So about this chapter, we're going to be dealing with primarily the autonomic division. We're going to talk about the specific neurotransmitters. We're going to talk about the basic sympathetic and parasympathetic responses. The autonomic nervous system is considered to be the quote-unquote involuntary nervous system. That is to say, heart, stomach, digestive tract, lungs, breathing rate. There are other muscles that are regulated by the autonomic nervous system, but those four organs in particular are the big ones. As opposed to the somatic nervous system, which is voluntary. So we're talking about skeletal muscle primarily when we talk about voluntary muscle movements. So what are the key differences? One of the key differences is what I would say is anatomic. That is to say, you have a somatic motor neuron originating from the central nervous system. Cell body would be located up here. The axon projects out. This is called the ventral horn. The dorsal horn is actually for sensory input, afferent division. So it comes out the ventral horn, and that axon moves down in the peripheral nervous system to its first synapse, which is usually at the effector organ. In the case of somatic, we're talking about skeletal muscle. So there's only one synapse between the central nervous system and the effector organ. In the autonomic division, and to a certain degree in the what's known as the enteric, which is almost a system unto itself. We're not going to split it out for these purposes, but in any case, what you have is again that ventral horn projection, the axon, and you reach what's known as a ganglion. It's an autonomic ganglion. It's an area where you have cell bodies and gray matter in the peripheral nervous system. So structures in the peripheral nervous system that contain a large number of cell bodies, somas, are known as ganglia. In the central nervous system, they call them nuclei. So we have this first neuron projecting out. We get to the ganglia, and we have one synapse. Then we have the neuron projecting out, and we have two synapses. So the difference in the autonomic innervation is that you have two synapses between the central nervous system and the effector organ. So we're going to look at the neurotransmitters, look at the different systems and what's going on in response to those signals. Some general functions of autonomic neurons is that they are controlling a lot of organs that are smooth or what we call involuntary. I don't like using the word involuntary because you can manipulate smooth muscle voluntarily. So let's say a combination of smooth and cardiac muscle. Lots of glands, like for instance, if you salivate, that's part of the autonomic neural function. Lymph nodes in some cases, endocrine organs, adipose in some cases. It's mostly involuntary, but not entirely. Rule of thumb, sympathetic mediates responses known as fight or flight. You are preparing for stress or you are engaged in highly metabolic activities. Parasympathetic, on the other hand, is what we would say rest and digest. You're storing energy, you're absorbing material in the digestive tract, secretions, and so forth. Okay? It does work hand in hand with the endocrine system, and it is modulated heavily by the behavioral state system. What that is to say, if you get butterflies in your stomach because you're nervous, that is activation of the sympathetic and parasympathetic systems to varying degrees in a time of stress. So it is closely related to these other systems, and sometimes I worry a little bit that by breaking them out, we forget how interconnected they actually are. Oftentimes, these systems are regulated through lower order neurons in the brain, through hypothalamus, and again, that's an interface with the endocrine system, pons and medulla in terms of muscle tone, autonomic function, etc. So what you have going on here is part of a reflex signaling pathway. You have some sensory input. That sensory input can be from sensory organs, or it can be from behavioral stimuli, leading to responses either in the hypothalamus, leading to endocrine responses or complex behaviors, autonomic responses. And so it is a highly interconnected system, that emotions and memories may affect other neural structures, and then leading to these responses. And that's why the responses can be quite complex at times. We're going to simplify it a little bit. Don't worry too much about that complexity, but I include this figure just to show you that level of overlap. This is a figure from your book as well. It's just showing you the various structures, that all of these structures tend to be in this lower order section of the brain. Memory, learning, leading to cranial nerves, particularly cranial nerve 10, which originates from this area of the brain. So you can see why they would be so interconnected. They're actually located close together in close proximity. Something that we're going to talk about here in just a moment is the idea that sympathetic and parasympathetic systems act antagonistically. It's a nice way to start. That is to say that they will do opposite effects in certain organs, and I'm going to give you an example of that in just a moment. But it is not entirely true. In fact, some of our most complex and nuanced behaviors are actually interactions of sympathetic and parasympathetic. But for purposes of this class, it's a nice basic paradigm. And so, as I mentioned in the previous slide, we talk about antagonistic control. For example, the heart is a classic example of antagonistic control. That is to say, if you have a sympathetic stimulation, the nerves will come down and activate these nodes, these pacemakers, to increase rate. Furthermore, the nerve will also go down into the myocardium and increase strength of contraction, modulates calcium flow. The parasympathetic, on the other hand, originates from the vagus nerve, cranial nerve 10, comes down and it innervates the nodes to slow heart rate down. But it doesn't normally affect strength of contraction, but it is slowing heart rate down. In fact, if you are sitting at rest, your heart is actually slowed down by the parasympathetic system from its base rate. If we were to sever these neural connections here, what you would see is the heart would sort of settle around 70, 75 beats per minute. But when you're quietly resting and relaxed, your heart rate settles somewhere between 60 and 70 beats per minute. And that's because the parasympathetic is actively dialing down heart rate. So that's an example of antagonistic control. Not always true, but it is the paradigm for our purposes. So here is a figure showing some of the basic responses when it comes to a sympathetic input versus a parasympathetic input. And I'm going to add a few entries to this. So this is sort of your key slide for effects. I will try to reinforce this with other figures as well. So some of the key responses are dilation of the pupils. So when you're under a lot of stress, your pupils, or you're stimulated sympathetically, I should say, not always stress. If you're stimulated sympathetically, your pupils will dilate. Your breathing will get faster and generally shallow, and you will see bronchodilation, that you'll see the smooth muscle in the lungs begin to relax and facilitate ventilation. We'll talk about that in a few chapters. The heart rate accelerates and with stronger contractions, and you will see reduced digestive activity, which is why when people are under a lot of stress, they often will have indigestion, because what happens is food material is sitting around in the digestive tract being processed by bacteria and fauna in the gut, creating gas, and so you'll often feel bloated or uncomfortable, unable to go to the bathroom, that sort of a thing. In addition, we would also see generally a vasoconstriction effect. That is to say, certain many blood vessels, particularly on the surface of the skin, but also in the gut, would constrict. And so this combination of increased cardiac output and vasoconstriction will lead to a general rise in blood pressure. And that should make sense. When you're stressed or when you're active, your blood pressure rises. On the parasympathetic side, your pupils will constrict. So it's kind of I find that sort of counterintuitive, that I would think that when you're calm, your pupils dilate, but it's actually the opposite. Pupils actually shrink during parasympathetic stimulation. Your breathing slows and deepens, and we'll see some bronchoconstriction. You will see some increase to a certain degree of the tone in the muscle in the lungs. So there is a bronchoconstrictive effect with parasympathetic. Your heart output slows down, so at decreased rate. Your gut becomes more active. Remember, rest and digest. And what we see a general vasodilation effect. So the slowing of the heart, the dilation of the blood vessels leads to a drop in blood pressure. And generally speaking, if it's rest and digest, most of the time, there are some overlaps here, but most of the time when you're talking about parasympathetic, you're also including increased secretions. And that can be in the lungs, the mucosal epithelia, digestion, and so forth. So that's a good list for you moving forward when you're talking about sympathetic versus parasympathetic. It'd be good to have a handle on this. With the autonomic pathways, sympathetic and parasympathetic, we do see some anatomical differences. For instance, sympathetic, what we have is various origins coming out of the spinal cord and then interfacing in what's known as a sympathetic trunk. I'll show you some pictures of that here in just a few moments. Whereas the parasympathetic, a lot of those are arising from relatively discrete areas of the brain in the cranial nerves. So from the brainstem, if it's autonomic, a lot of them are coming out of cranial nerve 10. So we have a slightly different anatomical structure to these systems. So with the sympathetic, like I said in the previous slide, you have various origins of sympathetic neurons, but they interface at what I would almost call a secondary spinal cord. There's one on each side, so there's one on this side, and we're not showing you the one on the other side. These are called sympathetic trunks, and they allow for lots and lots of interfacing of sympathetic neurons. That is to say, the sympathetic system is divergent and causing simultaneous responses across the body. And that's not only true in terms of the innervation, if you will, but we also get a significant amount of endocrine input here from the adrenal gland. So we're changing the behavior of some of these organs. In addition, we're firing off a little bit of epinephrine from the adrenal gland to reinforce that response. So we see generally widespread responses with the sympathetic. And that's due, in fact, to the divergence of the pathway. So you will see lots of interface in those sympathetic trunks. This is also where you see that ganglia located. That first sympathetic synapse is found in the sympathetic trunk. Here's just another picture. This one is actually showing you the sympathetic on the left side, okay, and the organs it serves. I'm not too worried about that. I'm more worried about the effects from a couple of slides earlier. Here's that sympathetic trunk, if you will, and that's where the ganglia are. You see the synapses there. On the other side, with the parasympathetic, you see the origins coming from either the cranial nerves. Okay, there are some exceptions to that, but what you see on the parasympathetic side tends to be more isolated responses that are not as divergent. Certainly this pathway right here is a bit divergent, But generally speaking, compared to the sympathetic, we have somewhat more isolated responses. And if I haven't already beaten this with a stick, this is just another recapitulation of the different responses. It includes a couple others, like for instance, sympathetic actually relaxes uterine muscle. It relaxes bladder muscle. Relaxes smooth muscle, but also slows it down, so you're decreasing peristalsis here. On the other hand, we're constricting blood vessels. Again, that first chart is more in line with what I'm interested in, but again, I like to reinforce, so I've included this figure. Sorry if it's confusing at all to you, please let me know. Now let's start getting down to the neurons themselves. As I mentioned very, very early on in this presentation, a motor neuron, a somatic system, has really only one synapse between the central nervous system and the effector organ, whereas the sympathetic and the parasympathetic have one and two synapses. They have two synapses on the way to the organ systems. We're going to show you some overlaps, some similarities, and we're going to break out the neurotransmitters. So we're going to spend a fair amount of time talking about the different neurotransmitters and the other molecules, other drugs that can affect these synapses. So as we drill down into the individual neurons of the autonomic nervous system, let's begin with the major neurotransmitters as well as the major receptors. One of the major neurotransmitters in the autonomic nervous system, but it's important all over the body, is called acetylcholine. The other primary neurotransmitter in the autonomic nervous system is called norepinephrine. It's a close cousin of epinephrine. In the UK, they would call it adrenaline and noradrenaline. So we have norepinephrine. It's another major neurotransmitter. A third one out there is dopamine, and we often hear about dopamine for Parkinson's, but we're really not going to talk about dopamine. We're going to limit it to acetylcholine and norepinephrine. Now, acetylcholine receptors are known as cholinergic receptors. So a neuron that would secrete acetylcholine is called a cholinergic neuron, and a receptor that is gated for acetylcholine is called a cholinergic receptor. And we have two major families of cholinergic receptors. One family is known as the nicotinic or sometimes nicotinic cholinergic receptor. Sometimes you see both words. Most often people will abbreviate it to just nicotinic. That is to say, these receptors are sensitive not only to acetylcholine, but are also sensitive to nicotine. Another family in the cholinergic family are called muscarinic receptors. That is to say, they are sensitive to acetylcholine, but they are also sensitive to a compound called muscarine, which is derived from mushrooms that were used for hallucinogenic and sedative purposes. So we have nicotinic cholinergic receptors and muscarinic cholinergic receptors, and we'll come back to those when we look at the individual pathways. On the norepinephrine side, as I mentioned, it's also known as noradrenaline. So the receptors for norepinephrine are collectively known as adrenergic receptors. So neurons that secrete norepinephrine are often called adrenergic neurons, and the receptors that are sensitive to norepinephrine are called adrenergic receptors. They may also be called adrenergic because they may be partially sensitive to epinephrine as well, to adrenaline. The two major families of the adrenergic receptors are alphas and betas. Alphas are found on certain organ systems. Betas and the subsets of betas are found on others, and they mediate different responses. But I'm introducing you to alphas and betas, nicotinics and muscarinics, because what you will find if you move on is that, especially in pharmacology, is that you will see drugs that are selective for these individual receptors. And even here, because there are a lot of side effects, if you gave someone a non-specific agonist for an adrenal receptor like epinephrine, you would see a whole host of responses that may be good or maybe not so good for the patient. So they worked very hard to create specific drugs for subsets of alpha and betas. I'm going on a little bit here, but I want you to understand why I'm taking the time with these receptors and why it will be generally important to you in the long run to have a handle on these receptors. So here are our neurons in the sympathetic and parasympathetic pathways. I'm going to work through this very slowly, showing you the neurotransmitters and the receptor populations. It's going to get a little bit complex visually, so I'm going to work very hard to work through this slowly and walk you through it. The first thing we need to do is identify where the acetylcholine neurotransmitter is functioning. So anywhere you see cholinergic, that's where acetylcholine is functioning. So we have acetylcholine in these three spots: in the first synapse of the sympathetic, in the first synapse of the parasympathetic, as well as the second synapse of the parasympathetic. A good rule of thumb, if you want something, is if you see the first synapse, and it doesn't matter whether it's sympathetic, parasympathetic, or even in the somatic, those all use acetylcholine, and they are all sensitive to nicotine. So the receptors in this ganglia right here, and in the synapse of the somatic motor neurons are nicotinic cholinergic receptors. So the first synapse is always a cholinergic nicotinic synapse. The neuron is secreting acetylcholine, so it's a cholinergic neuron. Sometimes you will see it referred to as a preganglionic neuron. So when we're talking about autonomic systems, if you see preganglionic, you know it's secreting acetylcholine. You also know that in the ganglia, those receptors are sensitive to nicotine. Now, I also showed acetylcholine down here in the second synapse of the parasympathetic pathway. Those receptors are sensitive to muscarine. So that means that this receptor here is muscarinic. So when we talk about sympathetic, we have a nicotinic cholinergic synapse. We have a nicotinic cholinergic synapse here in the first synapse. The second synapse in the parasympathetic is muscarinic. So if we were looking at a salivary gland or a smooth muscle and so forth, those receptors, if they're associated with parasympathetic responses, are muscarinic. Now the second neuron synapse in the parasympathetic pathway is adrenergic. So the postganglionic neuron is an adrenergic neuron that secretes norepinephrine. And the receptors on the target tissue for sympathetic pathways are adrenergic receptors. So let's walk through this. In the sympathetic pathway, we have cholinergic, nicotinic, adrenergic, adrenergic. Okay. For parasympathetic pathways, it's cholinergic, nicotinic, cholinergic, muscarinic. It seems complex maybe at first, but if you start working with this, it becomes quite simple. I always say, remember the first synapse, whether it's sympathetic, parasympathetic, or somatic, is nicotinic. The second synapse in the parasympathetic is muscarinic. The second synapse in the sympathetic is adrenergic. Easy peasy. So both the sympathetic and parasympathetics, the preganglionic neurons are cholinergic, and the receptors are nicotinic. Postganglionic sympathetics are adrenergic, norepinephrine. Okay. Postganglionic parasympathetic neurons are muscarinic. That is to say, the neuron is cholinergic, but the receptors are muscarinic. Work on that a little bit. You can work on that. It will become much easier and will save you huge amounts of time when you get to pharmacology. Just another picture of the same thing from your book. Okay? Both are cholinergic nicotinic at the first synapse. Sympathetic is adrenergic. Parasympathetic is a cholinergic muscarinic receptor. Okay? So good idea here. Good idea to practice these. You can really rock this if you just take a little bit of time and reflect on it a little bit. It's very, very powerful, especially if you're going to be involved in therapeutics. You know how when we looked at the anatomy of a neuron in Chapter 8, we talked about a presynaptic terminal? One of the things that we see in the autonomic system is we see a slight variation anatomically of that presynaptic terminal. That is to say, rather than a discrete presynaptic bud, if you will, single bud, smooth muscle, and when we get to Chapter 12 and talk about smooth muscle, I'll elaborate a little bit here. But oftentimes, smooth muscle, the muscle fibers themselves are not terribly well connected to each other. They're called multi-unit smooth muscle. And so in order to solve the issue of simultaneous contraction, the neuron has actually evolved to have this specialized presynaptic structure known as a varicosity. That is to say, you have these little string of pearls-like configuration, each one capable of secreting neurotransmitter, which means a more widely disseminated synaptic type of an interface, if you will. It still behaves like a synapse, but when we talk about single unit and multi-unit smooth muscle, understanding these varicosities may be helpful. So I've included that slide as part of this chapter. So these varicosities, if you will, are these sort of specialized structures. They're basically specialized presynaptic terminals that allow for a wide distribution of neurotransmitter secretion onto an entire smooth muscle. That's the main place where you see it. I would also like to roll in the idea of termination of signals. So remember in Chapter 8, we talked about exocytosis of neurotransmitter to initiate a signal across the synapse, and that we had termination of signal, and we had those three mechanisms. Remember, reuptake, enzymatic degradation, and diffusion. Well, the same story holds here, and in the interest of reinforcing that termination of signal, I'd like to talk about autonomic system. So the neurotransmitter, when it's secreted into the synapse between either two neurons or into an effector organ, one way of diminishment of that signal is diffusion. The neurotransmitter is diffusing out of that space between the two cells. Remember, this is extracellular. So it is a relatively open system, so diffusion occurs. You will also get enzymatic inactivation. So spoken another way, metabolism by enzymes in the extracellular fluid. So the synapse is between two cells. It's extracellular. And those enzymes will chop up and inactivate that neurotransmitter. A very common one is called acetylcholine esterase. And we should have seen that in Chapter 8, acetylcholine esterase. That's an example of an extracellular enzyme that metabolizes acetylcholine. A third thing that can happen is reuptake. That is to say, the neurotransmitter is transported back into the cells, often by forms of active transport. Whether it's the intact neurotransmitter or a piece of the neurotransmitter, like acetylcholine, we're taking choline back into the neuron to be recycled. But in either case, we're taking it back into the cells. That is similar to reuptake. Also, and this is one that I'm going to include just a little bit of an extension, sometimes, like in the case of norepinephrine, what happens is we will reuptake the norepinephrine, but then we will degrade it using an enzyme known as monoamine oxidase. It's similar to synaptic enzymatic degradation, only it's occurring inside the cells following reuptake. So if I were to sort of venture for a second, I'm going to ask you to imagine this. With acetylcholine, we break up the neurotransmitter in the synapse, and then we reuptake the choline. With norepinephrine, we reuptake the norepinephrine, and then we degrade it with monoamine oxidase inside the cells. So a little interesting side note from from what we talked about in Chapter 8. Again, trying to reinforce you to keep thinking about these concepts. These are very, very druggable targets, and there are many, many drugs that will affect these enzymes and reuptake systems. So now we've talked a little bit about neurotransmitters and receptors. I would like to take a bit of a step back and sort of put some of this together with Chapter 8. So I'm going to talk about neurotransmitter secretion, which is the initiation of signaling between two cells. Okay, so here's cell 1 right here. Here's cell 2. This may or may not be a neural cell. This one is the pathway for acetylcholine as well as norepinephrine. But let's review this Chapter 8 secretion mechanism. Again, the idea being this is the initiation of signal between two cells, particularly two neurons. So the first step is that we have an action potential heading down the surface of the plasma membrane of the neuron and the axon. We hit the presynaptic terminal, and you see the change in channel population here at the presynaptic terminal. That is to say, we depolarize because of that action potential, and that triggers calcium channels. Now, inside the cytoplasm of the neuron, there's very little calcium. Calcium is an important second messenger. So when the voltage triggers these calcium channels to open, calcium will flow in from the extracellular space and trigger exocytosis. So the calcium serves as a second messenger here. So the first message is the depolarization. That causes a change in shape of these voltage-gated calcium channels. The calcium flows in, triggers exocytosis. Okay? So that's the secretion of neurotransmitter. Neurotransmitter diffuses out into the synaptic space and may or may not interact with a receptor. Sometimes it will, sometimes it won't. It depends on concentration and depends on the other actions of the termination in the next slide that we're going to talk about. If the neurotransmitter does interact with the receptor, that will trigger some change in behavior of that target cell, whether it's a depolarization effect on the cell or it's some other cellular response, like, for instance, muscular contraction, that sort of a thing. So in any case, we have action potential, triggers voltage-gated calcium channels, calcium diffuses in, triggers exocytosis, neurotransmitter diffuses out. If it can interact with a receptor, will trigger a response in the downstream neuron. Work through it sequentially. The steps should make sense from one step to another. It's one of the more difficult mechanisms for students, in particular when you're talking about the termination in the next slide. So let's go to the termination step. So let's look at termination. This is the ending of the signal, and we're going to actually use acetylcholine. I know it's a little bit more complicated than the generic termination pathway, but I want you to use your pattern recognition skills to see where the similarities are between the generic and the specific. So this is specific for acetylcholine. So once acetylcholine is secreted by the presynaptic neuron using the mechanism we just described, it will diffuse into this synaptic space. It may or may not reach to the receptor. The reason I say that is even though we're showing it interacting with the receptor here, there are tons and tons of acetylcholinesterase molecules in this synapse. That's why you can start and stop muscular contractions so suddenly when you contract your muscles. Why? Because in the synaptic cleft, in this space, there are lots and lots of these enzymes, and they're actively breaking down acetylcholine into acetate and choline. So if an acetylcholine happens to make it to a receptor, it'll trigger a postsynaptic response, for instance, muscular contraction. In any case, though, we're actively breaking up or enzymatically inactivating acetylcholine. So that's our enzymatic inactivation. And what we're going to do is we're going to reuptake just choline. So this is reuptake here. It's a form of reuptake, only it is choline that's being brought in. The acetate diffuses out. So here we have reuptake, enzymatic inactivation, and diffusion all working together. Okay? So that's how we terminate acetylcholine in autonomic systems. Okay? Turns out many poisons like sarin gas and so forth are inhibitors of acetylcholinesterase. That's why I'm going to the trouble of doing this. I don't want to make it any harder than it has to be. But it is so important in our everyday lives and understanding what's going on with chemical weapons as well as drugs and so forth to understand this mechanism. So if acetylcholine is secreted, it may or may not interact with the receptor. If it does, it'll trigger a response. But mostly it's being broken down by enzymatic inactivation, where choline is being taken up. By the way, that's secondary active transport right there. I didn't want to say it, but I had to. The acetate diffuses out. So reuptake, enzymatic inactivation, diffusion. Now that choline here in this metabolic pathway is recycled. So we recycle it. We've added back onto acetyl-CoA and off we go. So you can recycle acetylcholine quite effectively. Now let's look at norepinephrine. Again, it's pattern recognition. Pattern recognition. So we have both the secretion and the termination showing in this figure. So here's our action potential coming down the axon. It triggers a voltage-gated calcium channel. Calcium flows in by diffusion, facilitated diffusion, and that triggers norepinephrine exocytosis. Norepinephrine is out in the synapse. Maybe it hits a receptor and causes a response. That's a G protein coupled receptor, but I'm not going to test you on that. In any case, a lot of the norepinephrine is transported back into the neuron by reuptake. That's step 6 here, but I just want to show it to you now. So here's our exocytosis. Okay. We get our interaction with receptor. That's step 4 in this figure. Understand, though, that these are all happening sort of simultaneously, if you will. So here's reuptake. That's step 6. Here's diffusion. That's step 5. Okay. These are happening at the same time. And then where's our enzymatic inactivation? Our enzymatic inactivation is up here in step 8. Step 7 is, well, we could repackage norepinephrine in some cases, or we're going to enzymatically inactivate it using monoamine oxidase found in the mitochondria. Again, action potential, voltage-gated calcium, exocytosis. That's the initiation. We have diffusion here, we have reuptake, and we have enzymatic inactivation right here. Step 4, step 7, not terribly important, not hard to remember. Remember, the point is to activate the receptor. Okay? We could have made this, any number of numbers. These are not necessarily sequential. Once we hit the synapse, we may have a receptor interaction, we may have diffusion, and we have reuptake, all happening simultaneously. But again, pattern recognition, diffusion, reuptake, enzymatic inactivation. I want to encourage you to have that flexibility. Just memorizing a given figure may serve a purpose for a point or two on an exam, but what is your goal? Your goal is to become an effective caregiver. And the more you can process this information, and maybe you can explain it to a patient if you need to. Maybe you can talk with a physician when you're talking about different types of therapies and options. As a patient, as a loved one, or as a caregiver. So don't be intimidated by these mechanisms. These ones are it's really good stuff and will give you a real edge when it comes time to either interview professionally or function professionally or deal with healthcare issues at any number of levels. So this slide I include just again to reinforce the idea that when you have epinephrine and norepinephrine involved in the signaling, they will cause different responses in different tissues that will be predicated on what type of adrenergic receptor they have. That the tissues do have discrete distribution of receptor depending on which tissue you're talking about. Like for instance, you see alpha 1s found in blood vessels, alpha 2s in pancreas, and so forth. The reason I'm bringing that up is because if you go further in your training, you're going to learn about these subtypes, recognize the responses, because we are more and more developing subtype-specific pharmaceuticals in order to avoid potential side effects. That's one of the big problems with drugs that are what we call adrenergomimetics. That is to say, drugs that stimulate the adrenergic system is side effects. Another little point here, this is just reinforcing something that we've talked about in previous chapters. I always like to bring material from previous chapters when I can. Look at what these norepinephrine receptors are doing. They're increasing or otherwise changing cyclic AMP, which implies that they are receptor enzymes. The enzyme that creates cyclic AMP is called adenylate cyclase. I'm not going to test you on that, is an enzyme. So when norepinephrine binds a receptor, it activates the enzyme and creates a second message. It hydrolyzes ATP into a circular molecule, that's what the cyclic means, cyclic AMP. Or in this one case up here, this is actually an enzyme that will cleave phospholipids. So if you have phospholipids in the plasma membrane, they serve as precursors to second messages. So it will cleave the phospholipase phospholipid molecule, and those cleaved products become messengers themselves. So it's an example of a receptor enzyme, the receptors for norepinephrine. In the case of muscarinic receptors, we see a similar story. In advanced classes, you'd memorize where these different muscarinic receptors are located, like in salivary or enteric or heart and so forth. Why? Because you would predict side effects or you would tailor the drug therapy for specific responses to avoid either dangerous or otherwise inconvenient side effects. Turns out many of these muscarinic receptors are G-protein coupled receptors. So you have the subtype here really tells you what type of 7 transmembrane module you have. In any case, what's going on is that the acetylcholine will bind the receptor and it will trigger a conformational change in this G protein, and that leads to second message. So it changes the shape of the receptor, that affects this G protein, and the G protein then causes downstream signaling in the cell. So it's a nice dovetail from our previous lectures, again reinforcing those concepts that we saw like in section 1, now applied in section 2. And now up to this point, we've primarily been talking about neurons, but I'd also like to take an opportunity to show you an interface between the neuronal system and the endocrine system. That is to say, the adrenal medulla and how it interfaces with the neurons of the sympathetic system. Now, just a little side note here that may help you down the line. When we talk about the middle of an organ, we often refer to it as the medulla. Okay, you see this in kidney, you see it in obviously here in the adrenal gland, but you can also see it in the brain as well and other organs. So when we talk about the middle, the medulla is actually a fancy name for the middle. Whereas the outside or the surface, the fancy name we use is the cortex, like the cerebral cortex, which is the outside of the cerebrum. Here we have the adrenal cortex, which is the outside of the adrenal gland. Whether it's the medulla, that secretes a certain set of hormones, and the cortex secretes another set of hormones. You might be familiar with corticosteroids, cortisol. That comes from the cortex of the adrenal gland. Our catecholamines that we've been talking about in this lecture arise from the medullary, the middle of the adrenal gland. So we have norepinephrine, like in the neurons, but we also have epinephrine. So let's show you how this interface works between the endocrine system and the sympathetic neuron. So let's work on this interface now. From the central nervous system arises a preganglionic sympathetic neuron, which is cholinergic. That means it's going to secrete acetylcholine in this synapse-like area. That acetylcholine will then activate nicotinic receptors on these specialized neurons called chromaffin cells. Chromaffin cells are derivatives of postganglionic sympathetic neurons. Postganglionic sympathetic neurons secrete norepinephrine. So like postganglionic neurons, they'll release norepinephrine when they're stimulated, only they will also release epinephrine as well. Turns out epinephrine's the major one. But in either case, they're being released and in hormonal fashion, in endocrine fashion, diffusing into the bloodstream and affecting target tissues. So it's almost identical to a sympathetic chain. Cholinergic, nicotinic. Adrenergic, adrenergic. So you can see a similar paradigm here as the neurons, only instead of going through a neural synapse, what we're doing is interfacing with a neuroendocrine cell, a chromaffin cell, and that in turn is releasing the catecholamines. That's why these catecholamines are called neurohormones, because these cells are modified neurons. So for our final slide, let's put these pieces together one more time. Remember, we're also going we need to know effects, sympathetic and parasympathetic, and some of the ancillary things like varicosities and so forth. But the primary goal was to get you to understand the structure and the neurotransmitters and receptors of the autonomic pathways. So let's start with parasympathetic and work our way down. Cholinergic neuron, acetylcholine nicotinic receptor. Cholinergic neuron, acetylcholine muscarinic receptor, leading to rest and digest. The sympathetic pathway, cholinergic neuron, acetylcholine nicotinic receptor. Adrenergic neuron, norepinephrine, adrenergic receptor, whether it's alpha or beta. Okay. The adrenal gland is cholinergic neuron, acetylcholine, adrenergic cell, we'll just say adrenergic chromaffin cell, adrenergic neuron-like cell, epinephrine, and a little bit of norepinephrine there, leading to target tissues through endocrine function. And that's it for this presentation. It is very much applied from Chapter 7 with the endocrine, Chapter 8 with neurons, where you have secretion and termination. Make sure you get to know those enzymes. Remember diffusion, remember enzymatic inactivation, remember reuptake. A little bit of anatomy when we talked about the structure of the neurons and the neural pathways. It's very much an integrated chapter with the concepts we've been talking about from Unit 1 and now Unit 2. I hope you've enjoyed this lecture. Good luck. Email me or contact me, and if you have questions or are confused about different aspects. Good luck, and I will talk to you next time.